Okay, good afternoon, ladies and gentlemen. Thank you for joining at APASL webinar. My name is Jin Mo-yang, Korea, presenter APASL. Today's topic is microbiota and liver disease. As you know, microbiota, which has been in the spotlight recently, is becoming increasingly important in our liver disease, such as development of hepatocellular carcinoma, acute chronic liver failure, hepatic encephalopathy, NASH, and so on. In particular, today's moderator, Professor Sarin, is the first to clinically adopted the relationship between microbiota and the fecal transplantation and liver disease in Asia. So I am looking forward to today's webinar. I will introduce today's webinar's moderator. As you are known, Professor Sarin is the Senior Professor, Hepatology and the Director, Institute of Liver and Biliary Science, ILBS, New Delhi. He's setting up the ILBS on the government of Delhi, and he is also the Director, WHO Collaborative Center in Chronic Liver Disease and Viral Hepatitis at ILBS. He has more than 600 publications to his credit, edited 13 books on liver disease, and contribute 87 chapters in various medical textbooks. He helped develop 19 major guidelines and the chairman of a steering committee of APASL and the editor of Hepatology International. And he got many awards. And I introduce the second moderator, Jasmine Bajaj. He graduated the Wisconsin Medical College. Now he has a professor of Virginia Commonwealth Medical University, USA. His concern about serious microbiota and hepatic encephalopathy. Also, he published many papers about microbiota and cirrhosis. So, Professor Sarin, please.
Thank you, President Jin Mo-yang from Korea, and I must compliment you and your team for having set up this very nice program and having invited me and Professor Bajaj to moderate this session. We are very lucky that APASL has and the workers in APASL have contributed to this area. The philosophy of using microbiota in different liver diseases is not new. It actually started from China and maybe Japan early enough. So now what we are doing is using these techniques in our patients. At ILBS, we have a fecal microbial transplant unit where we do almost regularly FMT in different groups of patients. One of our lead workers is also a speaker today, Dr. Shastri, who has joined us. We have an excellent team of speakers and a very excellent team of panelists today. I would also like to say that we are privileged to have Professor Bajaj, Jasmine Bajaj, with us, who has done stellar work in the patients with FMT. I am not seeing Dr. Bernd Schnabel. Is Professor Bernd Schnabel online somewhere? If you're on, can you please switch on your...
He's pre-recorded. It's 3 a.m. in
it's pre-recorded.
Right now. Yeah, it's 3 a.m. in California right now.
All right. So with that, maybe we would have the first presentation on gut-liver axis modulation in alcoholic liver disease by Professor Bernd Schnabel. Most of us know that.
Yes, we have video.
He has both experimental as well as clinical work related. Both experimental as well as clinical work related to biota.
Hello, I am Bernd Schnabel and I'm a professor of medicine and director of the San Diego Digestive Diseases Research Center at the University of California in San Diego. I would like to thank the organizers, the Asia-Pacific Association for the Study of Liver, and especially Professor Young Suk Lim for inviting me to talk about gut-liver axis modulation in alcoholic liver disease. These are my disclosures, and this is the overview. I would initially introduce the gut microbiome and the alcohol-associated liver disease. I want to then talk about changes that occur in the gut microbiota composition in alcohol-associated liver disease. I want to then switch over and tell you about the contribution of the gut microbiota to alcohol-associated liver disease. And finally, I want to highlight some precision approaches to restore host microbiome homeostasis to treat liver disease. As you know, if somebody drinks chronically alcohol, the patient will eventually end up with hepatic steatosis. However, the minority of the patient will advance to more progressive liver disease, including steatohepatitis, fibrosis, or cirrhosis. We have one special entity which arises predominantly in patients with an underlying cirrhosis and which is called alcoholic hepatitis. Alcoholic hepatitis still has a very high 90-day mortality, and there is currently no effective therapy available. The number of liver transplantations for alcohol-associated liver disease is rising, and the number of liver transplants for alcoholic hepatitis is also increasing, especially over the last several years. The human gut microbiota consists not only out of bacteria, but we have also fungi and we have viruses present. Especially the viral microbiota has the largest number of microbes in the gut. We have about the same number of human cells as we have microbial cells in our body. However, if we look at the gene numbers, the microbial genes outnumber the human genes that are present in our body. So the question is, does the gut microbiota contribute to alcohol-associated liver disease? And we know from a landmark study published by Gabriel Perlmutter in Paris in 2016, he transplanted stool from alcoholic hepatitis patients into germ-free mice, and then he fed these chnodobiotic mice chronically alcohol. And as you can see here, these mice transplanted with stool from alcoholic hepatitis patients had the highest liver injury and hepatic steatosis as compared to the mice that had the conventional microbiome after alcohol feeding. So suggesting that alcohol-associated liver disease is transmissible in mice. Does the gut virome change in patients with alcohol-associated liver disease? And we have recently published this study. And in this study, we isolated viral particles from patients with alcoholic hepatitis, patients with alcohol use disorder, and from non-alcoholic controls. And as you can see, the virome consists predominantly out of bacteriophages. Now, the most significant change that we found between alcoholic hepatitis patients and alcohol use disorder patients or non-alcoholic controls was the significant increase in mammalian viruses. If you just look at the composition of the virome, each patient is presented in one column. You can clearly see that the composition of the virome in patients with alcoholic hepatitis is different from alcohol use disorder patients and from control patients. If we look at the viral diversity, we found that the viral diversity was highest in patients with alcoholic hepatitis and lower in alcohol use disorder patients and lowest in control patients. Focusing more on the mammalian viruses, we found that parvovirity were significantly increased in patients with alcoholic hepatitis and alcohol use disorder patients as compared with non-alcoholic controls. In addition, we found a significant increase in herpes viridae, especially Epstein-Barr virus, in patients with alcoholic hepatitis, and we did not detect this in the stool from alcohol use disorder patients and non-alcoholic controls. When we looked at the MELD score in patients where we detected herpes viride, this subgroup had a significantly higher MELD score as compared to the subgroup of patients where we did not detect herpes viride in the stool. Switching over now to the fungi, and the question I want to answer is, does the gut fungal microbiome change in patients with alcohol-associated liver disease? In one of the earlier studies that we did, and this is an example from a mouse model where we chronically fed either an isocaloric diet or an ethanol-containing diet for 8 weeks. And we simply plated the feces on YPD agar plates and then assessed fungal colonies. And as you can see, in the control mice, there were hardly any fungal colonies detectable, while chronic ethanol feeding resulted in a significant increase in fungal colonies. We saw similarly in patients with alcoholic hepatitis a significant increase in fungal colonies when we cultured feces from these patients as compared with the alcohol use disorder patients and as compared with the non-alcoholic controls. We also did qPCR from single colonies using specific primers for Candida albicans, and you can also see that in patients with alcoholic hepatitis there was a significant increase in Candida albicans. We also used culture-independent techniques, in this case ITS sequencing, to characterize the fungal composition in the stool from control subjects, patients with alcohol use disorder, and patients with alcoholic hepatitis. You can see that in the control subjects, 1/4 about was Candida, shown here in red color, and then Candida essentially takes over in patients with alcohol use disorder and in patients with alcoholic hepatitis. When we look at the beta diversity, we could not discriminate patients with alcoholic hepatitis or alcohol use disorder, while they were clearly distinct from non-alcoholic controls. Furthermore, the fungal diversity was highest in the control patients as compared with patients with alcohol use disorder or patients with alcoholic hepatitis. So these studies suggest that the fungal composition in the gut microbiome is more or less driven by chronic alcohol abuse, but is less dependent on the liver disease stage. So the question is, how does the gut microbiome contribute to alcohol-associated liver disease? And I will just summarize many of the findings from the last several years. And this is a review we published also recently. You can see on top the intestine with the gut lumen, and then the intestine, as you know, is connected via the portal vein to the liver. So all the venous blood is drained via the portal vein to the liver. So whatever microbial molecules, metabolites, or other products are leaving the gut, the liver is really the first organ in our body to encounter all of these molecules. So what we learned over time is that pathogen-associated molecular patterns like lipopolysaccharide or LPS, but also beta-glucan from fungi, they can contribute to the progression of alcohol-associated liver disease. We also have learned that not only microbial products, but also live bacteria can translocate from the gut lumen to the liver and can contribute to alcohol-associated liver disease. We also know that indoles are decreased. They are very important tryptophan metabolites, and these are produced by the gut microbiota, and they're very important to induce, for example, antimicrobial activity in the intestine. Furthermore, we know that very important mediators of alcohol-associated liver disease are changes in the bile acids composition, but also in the amount of bile acids. Furthermore, we also have learned that short-chain fatty acids, for example, butyrate, are decreased, and that might also contribute to the progression of alcohol-associated liver disease. So in the following, I would like to highlight one of our own studies where we have shown how the gut microbiome, and especially bacteria, contribute to alcohol-associated liver disease and alcoholic hepatitis. In this case, we used culture-independent 16S rRNA sequencing and described the bacterial microbiome in non-alcoholic controls, alcohol-use disorder patients, and alcoholic hepatitis patients. You can see, again, beta diversity is distinct between the controls, between alcohol-use disorder, and between the alcoholic hepatitis patients. Bacterial diversity decreases from non-alcoholic controls to patients with alcohol-use disorder and further decreases in patients with alcoholic hepatitis. One of the most prominent changes that we found was a significant increase in Enterococcus faecalis in patients with alcoholic hepatitis. And these were about 2,000 to 3,000-fold higher in the alcoholic hepatitis patients as compared with the non-alcoholic controls. However, when we did correlation analysis between the total amount or relative abundance of Enterococcus faecalis with disease outcome and disease severity, we did not find any significant correlation. However, we found one toxin that is specifically produced by Enterococcus faecalis, which is called cytolysin. And we found that about 30% of the patients with alcoholic hepatitis are cytolysin positive in the stool. We did not find in cytolysin positivity in control subjects, and we only found one patient with alcohol use disorder to be cytolysin positive. When we further analyzed the survival in patients with alcoholic hepatitis, you can clearly see that the patients with cytolysin negativity had a very good survival, while about 90% of the patients with cytolysin positive stool that died within 180 days. So very strong biomarker to indicate a worse outcome and high mortality in patients with alcoholic hepatitis. In the last part of my talk, I would like to focus on precision approaches and how they can be used to restore host microbiome homeostasis to improve liver disease. So in the following, I would like to give you some examples how we can use the gut microbiome as a target for therapy to improve alcohol-associated liver disease. And the first example is that we can use bugs as drugs. So we can either use fecal microbiota transplantation to completely exchange the gut microbiota. We can use beneficial bacteria like probiotics, we can use prebiotics or symbiotics to add beneficial bacteria to an otherwise dysbiotic microbiota. Or we can use engineered bacteria to add to a patient's microbiota to produce beneficial products or to eliminate and metabolize toxins. I would like to provide some evidence that FMT is beneficial for patients with alcoholic hepatitis. In this case, a very small study coming out from India showed that fecal microbiota transplantation in steroid ineligible patients with alcoholic hepatitis shows survival benefit. These patients were treated for 8 days with a daily FMT via a nasoduodenal tube. In the FMT group showed a much better survival as compared to a historical control group. This obviously needs to be repeated and further better controlled, and this is a very small trial, so more patients need to be included. We know that FMT has also side effects, and one of these side effects was recently published in the New England Journal of Medicine, where drug-resistant E. coli were transmitted via fecal microbiota transplantation. Another concept that I would highlight, we can essentially drug the bug. And in this case, what can be done is we can either use, for example, antibiotics or antifungals that we eradicate broadly bacteria or fungi. On the other hand, we can use bacteriophages to selectively target now some of these bacteria and to specifically eliminate them. In the following, I would like to give an introduction how bacteriophages work.
The basic structure consists of a head, a sheath, and tail fibers. The tail fibers are what mediate attachment to the bacterial cell. The DNA stored in the head will then travel down the sheath and be injected inside the cell. Once inside the cell, the phage will hijack the cellular machinery to make many copies of itself. Lastly, the newly assembled phages burst forth from the bacterium, which resets their phage life cycle and kills the bacterium in the process.
And this is the most important part, that these phages are amplified inside the bacteria. Once they're released, their progeny can now affect new bacteria, and the process starts from the beginning. And we have used this now in a mouse model of alcohol-associated liver disease. So what we did is we used germ-free mice, and we colonized them with feces from cytolysin-positive alcoholic hepatitis patients. And then we treated these chnotobiotic mice with either a control bacteriophage or with a bacteriophage that specifically recognizes the cytolysin-positive Enterococcus faecalis, and then we subjected these mice to chronic ethanol feeding. And as you can see here, the mice that are treated with the bacteriophages against the cytolysin-positive Enterococcus faecalis, they have less liver injury as compared with the control bacteriophage treated mice after chronic ethanol feeding, and they have also less liver disease. So suggesting that phage therapy targeting the cytolysin-positive Enterococcus faecalis reduces ethanol-induced liver disease in this humanized mouse model. Finally, in this last concept I would like to highlight, we can also use drugs from bugs. So bacteria make very beneficial metabolites. For example, they can modify bile acids. They can also produce short-chain fatty acids. And these metabolites and products can be used to improve liver disease. So this is my key takeaway slide. Alcohol-associated liver disease is accompanied by changes in the gut microbiota. Intestinal microbiota contributes to alcohol-associated liver disease. And hopefully I've been able to convince you that intestinal dysbiosis represents an attractive target for therapy, but this needs to be personalized and requires precision approaches. Finally, I would like to highlight and advertise for our Gordon Research Conference on alcohol-induced end organ damage, which will be held in person between October 24th and October 29th this year in Ventura, California. This is between Malibu and Santa Barbara. I am organizing this together with Dr. Sabo, and we have secured Dr. Bruce Beutler as keynote speaker. He's a Nobel laureate from 2011. He discovered toll-like receptor 4. I would like to thank you for listening.
Okay, thank you, everyone. Thank you, Dr. Schnabel, for that excellent talk. We already are getting a lot of questions that are in the chat. If you do have more questions, please put it in the chat as well. The question and answer session will be at the end. I'm very happy to introduce our next speaker, Dr. Guangqiu Ko, who is a professor of medicine at Seoul National University, who's published extensively in NAFLD and NASH, as well as microbiome. He has a stellar record of service and training as well in Seoul, as well as Harvard and UNC Chapel Hill. And he's currently in the Division of Environmental Health Sciences. He's going to talk to us about microbiota and non-alcoholic fatty liver disease. On a related note, I would like to thank the organizers for giving this opportunity for us to moderate what is a very stellar session here. And with my co-moderator, Dr. Sareen, whom everyone respects, I think this is turning out to be a very nice educational opportunity and an interactive opportunity. So please send your comments and your questions via chat. Thank you. Dr. Ko, please.
Thank you very much, Dr. Bajai. It's very nice introductions. And I'd like to share my slide first. I hope you see my slide quite well. So my name is Kwang-pyo Ko at Seoul National University. And there is something I need to disclose. So I've been working on microbiota for a number of years. I've been trained as microbiologist for past 30 years. And for many diseases was affected by both our own genetics and the environmental factors, including the gut microbiotas. So the disease, something like NAFLD, is a chronic disease. It's more significantly affected by chronic exposure to the environmental factors, such as the gut microbiota. So, as you know, fat liver disease is caused by either excessive alcohol intake or the metabolic disorders. And I would like to share two studies with you for the next 20 minutes or so. That was recently published from my lab. The first study is using the Korean NAFLD cohort, collaborating with Professor Won Kim at SNU Brahma Hospitals. So we analyzed over 200 subjects on the Korean NAFLD cohort, which was the very precisely diagnosed with the biopsies. And when you analyze the microbiomes, we found out that the microbial community changes, according to the fibrosis severity, are quite different between non-obese and obese and NAFLD subject. When you look at this, all the population altogether, we couldn't find any, especially the patterns, and also we couldn't find any significant association in obese population. But we found that there is a significant association with gut microbiotas and non-obese population. And also, when you analyze the stool metabolites, such as the bile acid and the short-chain fatty acid, and number of different metabolites from the stool, and we found that non-obese subjects were also different patterns and significant associated with number of these stool metabolites with this NAFLD disease. And particularly the Ruminococcus and Veronella showed significant interaction with bile acid and in both populations. When you look at this more specifically on these particular families of these microbiotas, Veronella cells were positive associated with the increased fibrosis levels, and on the other hand, Ruminococcus were negatively associated in particularly among only non-obese population, with the difference, the criteria without the cirrhosis subject or the with non-NAFLD subject, only the severe fibrosis, the patterns was quite similar. And also, we analyzed the specific bile acid with the same categories. This is the first three is non-obese with the fibrosis levels, and obese population with fibrosis levels. We see particularly the bile acid such as cholic acid and chemodeoxycholic acid, what is positively associated with this one, and some of the short-chain fatty acid, particularly the propionate, was significantly associated with this the different fibrosis levels. So we have this gut microbiome profile used at the 16S ribosomal RNA sequencings and the stool metabolite. We believe that combination of these gut microbiome markers and fecal metabolite diagnose significant fibrosis with AUC of the 0.939 in non-obese subject. And the obese subjects was AUC was 0.520. And also, we performed some of the subject with the whole genome shotgun sequencing, and about the 38 the non-obese subject to confirm our those data. And we found out the BSH and BICD genes, which is to relate to the bile acid metabolism of the particular strain, were identified, and this was the significantly downregulated in non-obese subjects with significant fibrosis. So since our study was done with the Korean cohort, so we want to confirm that the different population, Western population, is this consistent with our research or not? So in this case, in Western NAFLD code, we download from the public database, and in conclusion, we find the similar patterns in the Western NAFLD cohort population. And also, when you identify these from the whole genome shotgun sequencing, some of the microbial species were improving the NAFLD disease. So whether this is only just co-occurrence or the causality, so we vigorously screened these our libraries, and then we identify Ruminococcus feces, and then we did the mouse experiment whether this is the causal effect or not. And the administration of this particular strain shows the improvement of the ALT and AST levels, and also, and in this case, the MCD diet models, and we also confirm these two different mice models, CDA-HFD models and DBDB models. We confirmed that the elevating effect of the Ruminococcus feces on diet-induced liver damage in 3 different NAFLD models, and also we believe this the gene related to this family is related to the different stool metabolite, which is the one of the very close association with the NAFLD disease. And I want to move on to the second study of our the talk. And a number of years ago, we published the particular strain of the gut microbiota between the healthy and the metabolic syndrome groups. The group has been investigating the effect of microbiota using the Korean twin cohort, which is over 3,000 the Korean twin population in Korea. So the advantage of using the twin is we can adjust for the human genetic factors, and then we can see the effect of the specific gut microbiota on the particular disease of our interest. In this case, we analyzed 655 subjects, and then we identify this number of the particular bacteria, the species with the metabolic syndrome. This is the data with the adjustment of the human genetic factors. Particularly the echinacea we identified in this study were confirmed by many other groups. When he published that papers, the Ruth Lay, he is at the Max Planck Institute right now, at the time the Cornell University, using this UK twin cohort. They published the very similar data. So in overall conclusion, the echinacea muciniphila has a beneficial effect on the metabolic disease. So our question was how this echinacea muciniphila can the effect on this metabolic disease. So we isolate 4 different strains of echinacea muciniphila from the healthy stool, and then we found out we basically did confirm this the administration of the echinacea muciniphila induced the weight loss and regulated the glucose homeostasis in this mice model. And also, the echinacea muciniphila modulate the brown adipose activation. So in this case, these in histologies and IBAS cells or number of cells and specific the IBAS specific gene expression has been changed after the administration of accumulation Eosinophilia. At the same time, it changed the body temperature on body temperatures, a slightly increase of the body temperatures and the M2 macrophages. And also we found that accumulation Eosinophilia induced the GLP-1 expression. So administration of these, the particular the microbial the species that increased the GLP-1 increase we find, and our question was, what is the molecular mechanism to show that specific the effect? So we did the cell-free spontaneous. We break down the cells and trying to identify the particular the fraction to induce the GLP-1 secretion in the from the accumulation Eosinophilia. And when you compare to the others, is known for this beneficial effect. We identify a particular fraction of the accumulation Eosinophilia that dramatically increase the GLP-1 secretion in this case. So we're trying to find the responsible for this the increase of the GLP-1 secretion. So we did the accumulation Eosinophilia. We separate the palate and culture spontaneous, and then we identify the fraction through these the FPLCs and LC-MS. We trying to identify the particular the molecules, and then we particular genes we clone to the plasmid and express the in the E. coli systems, and then we screen that, and then we identify a P9, the protein, is significantly, when you look at this y-axis, significantly increase the GLP-1 expression in NCI H716 and also a glutex cell line as well. So after this identified the particular strain, and we have to purify the P9 protein, and then we did the same experiment in animal models. So when you did the animal model, when you did the daily oral garbage of P9, the protein, we found the same effect, actually the better effect when you did the hyperdial models, and then the weight loss, and also the particular fat increase and glucose, the homeostasis was improved, and also we found the GLP-1 secretion as well. And also P9 induced thermogenic genes in brown adipocyte. And also we found this UCP1 was the expression was increased in this case. And so we find that the P9 was the improved the glucose homeostasis and the improved the metabolic disease, and also we find the P9 reduced the liver steatosis. So when you analyze in these hyperdial models, and this is normal chow and high fat, and the high fat is with the P9 administration, this improvement of the liver steatosis. So we try to investigate further what is the molecular mechanism. So P9 was released from the accumulation Eosinophilia. Accumulation Eosinophilia are living near the mucus, very close to the gut epithelium. So what is the molecules to recognize P9? So we did the ligand receptor capture technique based on for the triceps. We identified the binding, the potential bindings of the KTN1 and ICAM2. And when we did this in the gluten levels, and we did we administered P9 and increase of this GLP-1 secretion, and we confirmed that ICAM2 was responsible for this P9 interaction, because when you add the ICAM2 peptide, the inhibitions and the it was significantly reduced, and also when it blocked by the ACAM2 antibodies, and it's reduced by GLP-1 secretion was reduced. So we believe that the ACAM2 is responsible to interact with the P9 secreted by the Echinacea muciniphilia. And also, the expression of ACAM2 was not induced by P9. So P9 was not increasing the ACAM2, but we also find the IL-6 knockout mice was reduced. And also we confirmed that P9 was the administered P9 can slightly increase the IL-6 as well. So P9 regulates the glucose homeostasis in IL-6 dependent pathways. So when you did the when the glucose homeostasis when the wild types, we see the improvement of glucose homeostasis. But when IL-6 knockout mouse, this phenotype disappear. And also, we observed the same result when you administered by the Echinacea muciniphilia. P9 increased the lactate temperatures and GLP-1 secretion, and also through these IL-6 dependent ways. So in this case, the plasma GLP-1, the wild types, when P9 is increased, but the IL-6 knockout mouse disappears, and we could not see the lactate temperature increasement when you did the IL-6 knockout mouse. And so P9 is interacting with the ACAM2, and we want to further analyze what is the cell signaling related to these phenotypes. And when you did the RNA-6 the analysis, and we find out that some of these the cell the pathways responsible to calcium ions and response to cyclic AMPs levels increase. So we basically conclude that P9 increased the calciums and trapped attraction factors signaling pathways. So when you confirm in well lab, when you use the NCI-H716 cells, the add of the P9, the protein, to this cell increase the calcium level here, and also to increase the crab. So in conclusion, we believe this is the molecular mechanisms of the P9. So Echinacea muciniphilia release the P9 that interact with the ACAM2, and then it's it release the GLP-1, and it's affecting the immune cells, increase the release the IL-6, and and also the ACAM2 interaction increase the PLCs and calcium and the crab transfer factors, causing the GLP-1 increase, and thermogenesis in brown adipose tissues increased, and body weight decreased, and glucose homeostasis was improved, in order to improve the liver statuses. So those two recently our published paper shows that interaction between these obesities and the NAFLD and the NASH, and we believe the number of different factors, including the human factors and the disease phenotypes and the role of the microbiotas, is very important to causing the liver disease. So I'd like to acknowledge my the groups. The first study was done by Gilje Lee, and the second study by Hyoshin Yoon, who is my current and former doctoral student. And also I'd like to thank the funding agencies and the collaborators. Thank you very much for your attention.
Thank you, Dr. Ko. This was a wonderful talk, and going so deep into the mechanism of something that is troubling us day in and day out. Again, please put your questions in the chat. We will have a separate chat. We'll have a separate session afterwards. And now the next speaker is Dr. Shastri, who's an assistant professor at ILBS in New Delhi since 2014. He did his training in Jawaharlal Nehru Medical College in Ajmer, Rajasthan, and then DM in hepatology from ILBS under Dr. Salim. He has special interest in alcohol-related liver disease, gut microbiome, and liver disorders, as well as endoscopic procedures and management of emergencies. He's going to talk to us about fecal transplant in liver disease. Dr. Shastri.
Yeah, hello everyone. Thanks for the kind introduction. Yeah, it's my privilege to be part of this session, and sessions are really awesome. And I'll start my speech on fecal microbiota transplantation in liver diseases. Yeah, I have no conflicts of interest. Not going up. Liver disease is very well, liver lives in very close intimacy with the gut, and hence the diseases of the liver will affect the gut as well. So dysbiosis, which is less of the good bacteria and the abundance of the bad bacteria, is a well-known thing, and liver diseases are known to cause the dysbiosis by the virtue of its portal hypertension, reduced gastric acidity, impaired motility, local and systemic immunological dysfunctions, and the bile acid changes. The liver is known to cause changes in the gut microbiome. And similarly, it's also the other way. The changes in the gut microbiome also causes the pathogenesis of various and most of the liver diseases, and its role is very crucial. Who is first, the dysbiosis first or the liver disease first, is sometimes very difficult to say. And dysbiosis itself will cause intestinal oxidative stress, intestinal inflammation, leaky gut, and more bacterial products in the circulation, corpuscle activation, inflammatory response, systemic inflammatory response syndrome. The modulation by the fecal microbiota transplantation by FMT will be a promising therapeutic and also maybe preventive option in the management of liver diseases by acting at various stages directly and as well as indirectly. And yeah, this is the outline. I'll start with the role of FMT. I'll be focusing mainly on the available clinical data on FMT in the liver diseases. And firstly, the alcoholic liver disease, as we know, the lactobacillus and bifidobacterium and ruminococcus, they are the good ones. They are reduced in alcoholic liver disease, and the initial works and the most of the work in dysbiosis or gut microbiome is mainly done on alcohol-associated liver diseases. And there is also overabundance of the bad ones here. And it has also been shown that some of the bacteria, are their abundance proportionately increases with the severity of the alcoholic hepatitis, which is the most florid form. And this is pictorial representation of the same. There is a disruption of the tight junctions. There is dysbiosis with lower lactobacilli, leaky gut, local inflammatory cells activation, and the liver inflammation. And it has also been shown both in the mice and the human studies that the bacterial overgrowth of the dysbiosis in liver diseases, especially the alcohol ones, are more pronounced in the proximal bowel. And as I said, the alcoholic hepatitis is the most florid form of the alcoholic liver diseases. Is very much in discussion because of the lack of its specific therapies available, and only standard of care, that steroid, which has only short-term survival benefit up to 28 days, and the longer-term benefit is not known. And even in those who receive steroids, there are a portion of patients who will not respond to steroids, and there is a larger chunk who are not eligible for steroid therapy to begin with. And additionally, those who do not respond and those who are ineligible, most of them also do not have transplant options because of the recent alcohol intake and other many social issues. So the treatment options are very minimal in this group. And as gut dysbiosis is the core or the central role in the pathogenesis of alcoholic liver diseases, so the modulation of this would definitely be a very appropriate way of tackling these patients and to cover most of the patients, not just confined to the eligible or ineligible. So this paper was discussed very well by the Professor Schnabel. It's a very interesting and very intriguing. The alcoholic hepatitis is transmissible. So the gut microbiome from the alcoholic hepatitis patients transferred or transplanted into the ethanol-fed mice, germ-free mice, produced liver injury with transaminases and increased fat content. And again, it was reduced by the transplantation of the microbiome from the stable alcohol patient into the injured mice, and it reduced. It shows that the alcoholic hepatitis itself is transmissible, and it is also curable by the good microbiome. And we need both alcohol and intestinal gut microbiome to act synergistically to induce alcoholic liver disease. And this was a second very interesting study published in JHEP17. Again, group of mice were adapted from the same facility and to different two labs, and one lab had alcohol sensitive mice and the other were alcohol resistant. The injury did not happen despite alcohol consumption. And it was found that the good bacteria like bacterials were high in the alcohol resistant mice, and whereas the sensitive mice had more of the bad guys, and it produced more of inflammatory cells, IL-1, IL-T, and other things. So what they did further was it came to be known that it's the microbiome that is reason for the sensitivity of the alcohol. What they did was they transplanted the gut microbiome from these mice, the resistant mice, into the sensitive mice, and this FMT reduced the ALT or the liver injury in the sensitive mice as well. So the FMT can cure alcoholic liver injury. That was the concept of this study. And based on this, this is the only pilot study available, human study on patients with severe alcoholic hepatitis. I'm happy to be part of this study. This group did FMT, fecal microbiome transplantation, in patients who were not eligible for steroids. That means they were really sick to begin with very high MALD of 31. And FMT was given through nasoduodenal tube on daily basis for 7 days. Exact technique I'm not going into details because constraints of the time. And 8 patients that were compared with the retrospective cohort of 18 comparable cohort with standard of therapy. And what was found was there was increase in the bacterioids and decrease in the proteobacteria in the patients who received FMT compared to baseline by repeat sampling done at 3 months. And there was also increased relative abundance of the bifidobacterium. Clinically, the survival was much better. The 3 month, 6 month survival was much better in the group which received FMT compared to the retrospective comparable cohort. So this led us to think that why not, as steroids has only short-term survival benefit, why not try FMT head-on-head with steroids, because it gives long-term survival benefit. This is again a study by Apurva. He did the study by comparing steroids compared with the fecal microbiome transplantation head-on-head for 7 days. And then steroids were managed as per the protocol, what we use standardly, and the FMT was given as we used in the previous study, 7 days daily and then follow it up for later. And this is unpublished data which shows that the FMT had better. This is the purple line is the FMT survival, which was much better than the steroid non-responders, and at par with the steroid responders till day 60. Beyond day 60, the survival was better than the steroid responders. And the numbers were 90, 45 in each of the limbs. And survival was in FMT limb around 84%, 90-day survival, transplant-free survival, whereas it was 70% in the steroid responders. So these are encouraging results which make us believe that the dysbiosis can be cured, and this can help in curing the most florid liver conditions like alcoholic hepatitis. So where do I place the FMT in the current treatment algorithm of alcoholic hepatitis? Basically is patients who are ineligible to steroids or not responding, or who have continued higher MALD beyond 30 days, if MALD is more than 14, or if the patient fulfills the criteria for transplant even after a month or 2, then also we can think of FMT in such patients, especially in those who do not have transplant options. So And this is the only available data so far for alcoholic hepatitis. And one more interesting data from the Professor Just Bajaj group, that this is the phase 1 trial double-blind RCT. This is on the problem drinking with audit score of more than 8 and cirrhotics, alcoholic cirrhotics. Number 20, 10 in each limb, and the FMT limb received FMT enema single dose on day 1 and compared to placebo enema in the control group. And what they saw was the alcohol craving questionnaire showed decreased craving in the group which received the FMT compared to the placebo enema group. Craving was reduced by 90% in the FMT group compared to only 30% in the placebo enema received group at day 15. And also there was reduction in the urinary ethyl glucuronide levels. And additionally, there was improved cognition, and psychosocial sickness impact profile was better in patients with FMT group. So this sounds good, that even the craving and the relapse and lapse can be prevented by using FMT in this group of patients. Second set of diseases in liver diseases, the second set of patients are the hepatic encephalopathy, where we have a lot of data on gut microbiome and the fecal microbiome transplantation, especially from the Professor Bajaj group. And it is well known that the dysbiosis ratio, mainly the lactobacillus ruminococcus, which produce the short chain fatty acids, are drastically reduced in patients who have end-stage liver disease and recurrent hepatic encephalopathy. And there is also overabundance of the urease producing or ammonia generating organisms in the patients with end-stage liver disease. These bacterial products and the like ammonium, mercaptans, and other things, in addition to the direct bacterial contents, the endotoxins and TNA, they directly trigger the inflammatory response and cause astrocyte swelling and pathogenesis. They have the crucial role in the pathogenesis of the hepatic encephalopathy. Thus, FMT can be a promising tool in addition to the standard of care, which is lactulose and rifaximin, by modulating the gut microbiota in patients with hepatic encephalopathy. Let's see the data what we have right now. This was the first case report published in 2016 on a ALD plus HCV 57-year-old guy with persistent or recurrent grade 1-2 HE. He was given 4 shots of fecal FMT. First shot was through enema, and the rest were first one was through colonoscope, and remaining were through the retention enemas. And the patient had persistent lethargy, reversal of sleep, and disorientation. His performance improved post FMT after 15 days, and which remained improved till a couple of months to come down, with the improved reaction time on the Stroop test, reduction in the serum ammonia levels, improved quality of life. So this was the first study which showed that FMT can help in hepatic encephalopathy as well. Based on this, the Professor Bajaj et al. Has published 2 very nicely conducted trials. The first one was the open label RCT, where FMT plus standard of care, the lactulose and the rifampicin, was compared with the standard of care without FMT. A single donor was used based on the microbial richness of the Lactobacillus and Ruminococcus. Single dose of enema was given after the broad spectrum antibiotics of 5 days, and they could see improvement in the cognitive tests, PFS and the Stroop test. And additionally, there was long-term good impact over 12 to 15 months with very low hospitalization rates, and the total number of HE events were almost null at 1 year follow-up, more than 1 year follow-up in patients who received FMT. This was a very small pilot study, 10 patients only, but still it's a wonderful result. And the second study they did was again the safety trial pilot study, phase 1 study. In this, they used the capsules containing this enriched bacteria, mainly FMT. 15 capsules were given in patients with again recurrent HE, and the number of HE episodes and hospitalizations or emergency visits were reduced drastically in patients who received FMT capsules compared to the controls. Additionally, they could also see the reduction in the inflammatory markers like interleukin-6, improvement in the gut microbial antimicrobial protein. They have done repeat biopsies, duodenal and sigmoid biopsies in the patients who received capsules after 3 months, and there was improvement in the antimicrobial proteins and reduction in the serum LPS binding proteins in these groups, and definitely there was also improvement in the cognition response in these patients. So there are many more trials in the ongoing trials registered in the GOV, and various trials are mainly concerned doing with the capsule versus enema, number of days of installation, dosing, especially lyophilized capsule versus frequency, and the route of administration, enema versus gastroscopic, and the long-term management. They have been studied, and it would be interesting to look at the results in coming days. So the next presentation in liver disease would be the NAFLD ones, the non-alcoholic fatty liver disease ones, and definitely this is one of the diseases where the pathogenesis is centered around the gut and dysbiosis. It has been shown that the butyric acid producing short chain fatty acid producing firmicutes are reduced in patients with a NAFLD, and additionally there is overabundance of these bad guys with along with the mainly the ethanol producing E. coli and Klebsiella are enriched in these patients with NAFLD. It is called auto brewery syndrome. So the concept is giving this magic pills for FMT will decrease the chances of NAFLD progressing to NASH, NASH progressing to cirrhosis by decreasing the gut inflammation or gut dysbiosis. So what is the data we have on clinical side? There are only 2 studies which are published so far. The first one is the double-blind, both are double-blind randomized controlled trials. The allogenic FMT in 15 patients versus autologous FMT in 6 patients, single endoscopic FMT instilled into the duodenum. They wanted to look at the HOMA-IR, the insulin resistance, and the liver fat through MR-PDFF after 6 months, but there was a failure of the results here at the primary endpoint. There was no change in the insulin resistance, and as well as there was no decrease in the hepatic PDFF fat content at 6 months. But the interesting part was they could see significant changes of in the small intestinal permeability to mannitol and lactitol. Mannitol, the intestinal permeability was reduced after 6 weeks. And in the second study, lean vegan allogenic donor was used, single donor was used in 10 patients compared with the autologous FMT in the control group. In this, they used 3 times at 8 weekly interval, 8 till 24 weeks. They give fresh nasoduodenal samples, and they did biopsy at baseline and after 24 weeks. There was a trend towards the reduction in the necroinflammation on histology and follow-up, and along with the there is a reduction in the mean ALT by -14. Although it was not that significant, there was a trend towards a significant reduction in the necroinflammation and ALT levels. This is the only data available in NAFLD. Many trials are undergoing, which have planned like many phase 1, phase 2 trials, open label trials. They are planning to do pre and post biopsy proven NASH patients, and upper GI route. Most of them are using the upper GI route, and I'm sure the results will be interesting to look at. And coming to the another interesting area, hepatitis B virus related and dysbiosis. This is very interesting to know that with in patients with hepatitis B, we all know that the immunology plays the major role. The change in the immunology leads to the activation of the virus and viral clearance or the disease progression. So there are various studies to show that the gut microbial changes are there in chronic hepatitis B patients and even in the mice. There are I'll not read the bacterial names, but the interesting part is with even in the HBV infected mice, once the entecavir is started or treatment is given, I'm sorry. Yeah, the dysbiosis reverses after the HBV infection treatment. And in patients with HBV, in addition, the only infection, hepatitis or cirrhosis, there is progressive reduction in the alpha diversity of the gut microbiome. So the gut microbiome having main role in the immunology, it helps in the activation of immune cells such as T cells, reduction of the inflammation, interleukins, and other things. So this is known to have an effect on the hepatitis B. There are a couple of studies, in fact 3 studies. 2 studies based on in patients who have been on long-term antivirals, E positive, non-cirrhotics with persistent E positive status. And it's interesting to see that the HBeAg levels go down after the FMT single dose, along with the antivirals, of course. There is reduction in the HBeAg levels, and the HB like you can see 4 out of the 5 patients who received FMT with E positive status lost their E antigen within 40 weeks. That is very interesting. But there is no HBsAg seroconversion. HBeAg decline and seroconversion was noted. In the second study done from AIMS Delhi, they have also looked at the similar group of patients, E positive, 14 patients. 2 out of 14 became E negative, which at 1 month and 3 months respectively after FMT from a single donor, single healthy donor. But these 2 patients had DNA undetectable at the start of treatment, but still they were E positive. And these are very preliminary results, but they are definitely encouraging. And the third trial, this was done at our place. Our group, we included the sicker group of hepatitis B, HBV reactivation. The mean MELD was 28, and we did FMT daily for 7 days versus along with tenofovir versus tenofovir itself alone. 32 patients in each limb. The 90-day transplant-free survival in this group of HBV reactivation ACLF was significantly different, 75% versus 37.5% transplant-free survival. Along with that, there was lesser incidence of infections, SBP and pneumonia in patients who received FMT compared to those who did not. And coming to the last part of the study, these are the interesting new roles for liver disease. These are the very interesting areas. Recently published one single RCT on PSC in FMT, FMT in PSC in fact. It is well known that there is reduced bacterial diversity and 3 bacterial genera, Enterococcus, Fusobacterium, Lactobacillus, they are over abundant in patients with PSC, independent of the fibrosis or IBD or UDCA use or liver transplant. So this group in 2019, they included the patients with PSC and IBD. It was an open label pilot trial of 10 patients, single endoscopic FMT in the right colon, single donor, no antibiotics used. There was reduction of alkaline phosphatase by more than 50% in 3 out of the 10 patients. And additionally, there was also improvement in the bacterial diversity in 1 week. And additionally, what they could see was the increase in the bacterial diversity correlated with the reduction in the alkaline phosphatase levels. So the bacteria were actually responsible for those who had a response in bacterial diversity, they had reduction in the alkaline phosphatase. And this is the only study available, and there is no studies available in the autoimmune hepatitis or PBC. And coming to the transplantation, this is an interesting paper from Tokyo. This is in patients with stem cell transplantations having acute graft versus host disease of the gut with bloody diarrhea and hematogesia, basically acute rejection, graft versus host disease. They gave FMT for 4 stem cell transplanted patients with acute graft versus host disease of the gut, and out of them 3 were steroid resistant or 1 was steroid dependent, and 3 patients showed complete response after the single dose of FMT through the duodenum, gastroduodenal tube, and 1 had partial response with reduction in the diarrhea or bloody diarrhea levels and even the engraftment. And they also could look at the regulatory T cell response to FMT in this particular group of patients. So possibly in our liver transplant as well, if to reduce the rejections or maybe to increase the acceptance, FMT might have a crucial role. And only data I could get hold on in liver transplant patients is this one. And it has been seen that after 21 days after liver transplant, there will be sudden increase in the diversity of the gut microbiome. With if you divide between the groups who develop acute cellular rejection and who do not, those who develop acute cellular rejection have these bacteria overgrown after 3rd week: Bifidobacteria, Streptococci, Enterobacteriaceae, and Bacteroides. In the non-ACR groups, the other ones like Ruminococcus, Clostridia, these families they overgrow, they are outnumbered. So possibly the transplant graft acceptance and the rejection will depend on the gut microbiome as well. So and this is another simple case report of a successful FMT in a patient of a post-liver transplant patient who had severe complicated Clostridia repeated infection, not responding to even vancomycin. They have shown that FMT first dose given per rectum, because it was very friable, next oral capsules were used. There was remission of the Clostridium difficile infection even in post-transplant patients who received FMT, and also it was safe even in post-transplant patients who received immunosuppression. So there are definitely many hurdles for FMT, and way ahead is not like it's one size doesn't fit all. and it is very difficult to find out the ideal donors. If the donor has a pro-inflammatory microbiota, the purpose will not be served. It is important to identify donors with good anti-inflammatory microbiota in them. And additionally, if the patient has a vaccine-winning disease like IBD with intermittent flares or HBV with reactive, the inactive disease timed gut samples or stool samples can be biobanked and used whenever there is a pro-inflammatory flares later. So banking of the autologous FMT can also be planned. So ideal route and frequency, it can also be, it is also not universal that this route is good or this is bad. It should depend on individual disease and situation based which route should be used and how frequently. We need more data to decide how frequently and how long. And it should be customized and tailored for individual patients according to his own microbiome, microbial signature. And definitely the durability of the desired effect is very important to be assessed in the future studies. And we should also be aware of the risks of unknown infections or donor phenotypes, like transmitting obesity to a person who was lean before FMT because he just received FMT from an obese guy. We should also be open to the unknown risks which can be associated with FMT. So to summarize, gut microbiota is involved in the pathogenesis of many and most of the liver diseases, and FMT is definitely a way to modulate the microbiota. Early but encouraging results in many liver diseases are available so far, and definitely FMT is a palatable medicine now, at least more palatable, and we need more good quality trials and very well conducted multi-center trials to prove or make it better utilizable to and reachable to many patients who may definitely see better life with FMT. And I would like to say thank you for the organizing committee for having me here, and thank you.
Thank you, Dr. Shastri, for this comprehensive presentation. In the interest of time, we have many questions, but maybe we can have them in the discussion part subsequently. And I would take the opportunity of introducing my dear friend and respected friend, Dr. Robert Schwab, who has spearheaded the area for a very long time. And he is an associate professor of medicine and also one of the pioneer workers in the field of liver regeneration and of course microbiota. And Robert would be speaking, I guess it is early morning time for him with coffee. He will be speaking on the role of microbiota in hepatocarcinogenesis. Robert, over to you. If this is a video, we may start, but I think we have Dr. Robert Schwab also available.
Yes, it will be a video.
Start the video.
It will be a video.
Certainly.
My name is Robert Schwabe, and I would like to thank the organizers for giving me the opportunity to talk today about the role of the microbiota in hepatocarcinogenesis. This is a rapidly growing field. A search of PubMed with the keywords cancer and microbiome has shown that there's virtually no publications in the early 2000s, but we have now an exponentially growing field with thousands of papers per year. Whereas initial studies were focusing on specific pathogens associated with cancer development, especially bacteria such as H. pylori driving gastric cancer and malt lymphoma, the field has transitioned and evolved to study the commensal microbiota and dysbiosis in carcinogenesis and also in cancer therapy. And starting out in colorectal cancer, this has expanded to virtually all tumors, shown roles of the microbiota in lung, breast, pancreatic cancer, and today's topic, liver cancer. The microbiota, I would like to emphasize that it's not just bacteria. I'm not going to focus on this in today's talk, but there has been a role of the microbiome in cancer development shown in pancreatic cancer, as well as in tumor therapy. I'm going to talk about 3 topics today. What is the evidence for the role of the microbiome in liver cancer? What are the involved pathways? And is the microbiome a therapeutic target? This is going to be an overview of the topic from multiple studies in the field. I'm going to weave in some of our data also, but these are studies that are mostly a few years old. We have transitioned since then to other topics, which still involve bacteria, but more as a therapeutic vehicle. So what is the evidence for the role of the microbiota in liver cancer? We know that there's dysbiosis throughout the disease progression from normal liver to different stages to HCC, with dysbiosis and increased translocation for 20 to 30 years, driving, for example, in fatty liver disease, the energy nutrient extraction and also inflammation. A study from the group of Jeff Gordon elegantly showed the role of the obesity-associated gut microbiome having an increased capacity for energy harvest. In addition, in later stages, the microbiome affects epithelial injury and stellate cell activation. We've shown a role for TLR4 in the microbiota in stellate cell activation of liver fibrosis. The same pathway is also ongoing in advanced stages of fibrosis, and in the transition from cirrhosis to HCC, 90% of tumors develop in cirrhotic livers. Inflammation, epithelial proliferation, survival play roles, as well as an increasing role of the immune responses in this setting. And this is the most relevant setting as most tumors develop in these patients with cirrhosis, and that's also going to be the main focus of the talk today. So most of the evidence comes from mice, especially functional studies, showing data from our 2012 cancer cell paper, where we first used antibiotics, got sterilized mice by a quadruple antibiotic cocktail, which was highly efficient at reducing the bacteria. And in a carcinogen-driven model, we could see a strong reduction of HCC development seen by multiple parameters in the group that received the antibiotics. However, to exclude that these antibiotics can directly affect the tumors rather than the microbiome only, although most of these are non-absorbable, we also studied germ-free mice, and to make a long story short, we saw a similar, very profound reduction of cancer development in germ-free mice. Similar results have been obtained by other groups and in other models. Here an example from the group of A.G. Hara, using a high-fat diet model of HCC, where they first inject a carcinogen, followed by high-fat diet, and this leads to profound tumors, which could be strongly reduced by antibiotics, either a quadruple cocktail similar to ours or vancomycin alone. And then finally, another example, more recent example, from the group of Vijay Kumar, where they used dysbiotic mice that were fed a diet high in fermentable fiber, and the metabolites from the microbiota drove the development of liver cancer in this model. So what is the evidence in patients? There are very strong data for showing dysbiosis in cirrhosis, which is a precancerous conditions, as 90% of HCCs develop in patients with cirrhosis, and HCC is the main cause of death in patients with compensated cirrhosis. And here is a comparison of cirrhosis with multiple diseases, not of the liver, but other diseases, such as colorectal cancer, inflammatory bowel disease, obesity, type 2 diabetes, showing the most profound fibrosis in cirrhosis. And multiple studies have shown very strong dysbiosis in cirrhosis, which can also be leveraged as a diagnostic tool. So how about dysbiosis in HCC? This is a study I'm showing you here comparing NAFLD-induced cirrhosis patients with and without HCC to each other, as well as to healthy controls. And the author showed a strong reduction in diversity between the healthy and the cirrhotic patients here, but there was no difference between cirrhosis with and without HCC. Similar data in the PCA plot here. However, taking a closer look at the genus level, the authors could see differences between cirrhosis with and without HCC. And they could also associate specific changes such as increased Bacteroides and decreased bifidobacteria and Akkermansia to changes in cytokine levels, inflammation, and immunity. One additional study here comparing early HCC to liver cirrhosis and control, and they couldn't really show consistent changes, at least in terms of the HCC. So there was a decrease in richness between the cirrhosis and compared to the control patients, but there was no a decrease, but even an increase in the early HCC. However, again, taking a closer look, the authors saw differences between cirrhosis and early HCC at the phylum and genus level. Here, at the phylum level, comparing cirrhosis to early HCC and control to early HCC. And here at the genus level, the differences. Here, the authors could also use a microbial signature to differentiate between HCC and non-HCC. A number of additional studies also showed changes. Here, one study showing an increase of E. coli in patients with HCC. A study comparing cirrhosis to HCC of mixed etiology found moderate microbial changes again. A study showing HCC of HBV and non-HBV origin, showing differences between those groups in terms of the microbiome, but again moderate. And another study on NAFLD cirrhosis and NAFLD HCC showing moderate changes in the microbiota. So overall, the differences between HCC and cirrhosis seem small to moderate, and they're not always consistent between all the studies, and much smaller than the differences between normal and cirrhosis patients. Again, keeping in mind that cirrhosis is a precancerous condition and could affect hepatic carcinogenesis. The functional relevance of these changes in patients with HCC seem unclear, and they need further investigations in people. A new additional topic I would like to introduce is intratumoral bacteria, which have been shown in a number of tumors, not only colorectal and gastric cancer, but additional tumors such as breast and pancreatic cancer. And there is increasing evidence for a role of intratumoral bacteria in HCC, a study profiling tumor-associated microbiota in HCC here, and another study showing a role of the liver microbiome, not specific in the tumors, being implicated in cancer prognosis in HCC. So this is an evolving topic, and we need further studies to characterize the intratumoral microbiome as well as the liver microbiome in HCC. The question arises whether these are live and proliferating bacteria or just bacterial remnants, and their role versus that of the intestinal microbiome to be further investigated. In the second part, I want to talk about pathways. I'm going to keep this rather short, as there are multiple reviews and also a number of original papers on this topic. So a main driver is the dysbiosis, as well as the increased translocation. Dysbiosis driving some of this. There's bacterial overgrowth also in the upper GI tract, and there are altered metabolites, bacterial metabolites that can also reach the liver. And altogether, this changes inflammation, metabolism, proliferation, fibrosis, and immunity in the liver, which can set the stage to liver cancer development. This is a slide taken from a recent review that I wrote together with Tim Creighton, showing a role of the leaky gut and dysbiosis, again, which are connected to each other. The leaky gut inducing the release of or translocation of microbe-associated molecular patterns, MAMs, are often also called PAMs, which activate pattern recognition receptors such as toll-like receptors on various cells in the liver, which include stellate cells and macrophages, driving inflammation, fibrosis, the senescence-associated secretory phenotype, and the release of mitogens such as epiregulin, which can drive hepatocyte proliferation. A recent paper also showing a role for TLR4 and hepatocytes driving the activation of stellate cells and fibrosis. So altogether, these can create an environment which is more prone for the development of HCC. Dysbiosis and bacterial metabolite can additionally change this environment. Metabolites such as secondary bile acids acting on stellate cells, L6 and other cells, also influencing hepatic immune responses. Metabolites such as TMA, which is also acting on and metabolized by hepatocytes, turning it into TMAO, affecting the environment, as well as ethanol, which can be produced by bacteria. And as mentioned before, bacterial metabolites can be converted to short-chain fatty acids, and also these diets in dysbiotic mice that are high in soluble fibers and these bile acids together, these metabolites can then drive the development of cancer. In the third part of my talk, I want to focus on the microbiome as a therapeutic target in liver cancer, and first talk about cancer prevention. As mentioned before, there's chronic dysbiosis over a long time in different stages of the disease, and so we could target the gut-liver axis at different stages and time points. However, I would argue that targeting in these early stages, we needed to treat a lot of patients to prevent development of liver cancer, so it probably makes sense to consider this targeting only in late stages of the disease. Related to this, I would like to show you again some data from our cancer cell paper, where we used non-absorbable antibiotics and treated mice at early and late stages. And to our surprise, at early stages, we only saw a moderate reduction of cancer development, whereas when we treated the mice late in the second half, we saw a profound reduction of cancer development, also seen here by various parameters. So this indicates that antibiotics could help to prevent or delay HCC development in patients with advanced liver disease. How could this be achieved? There are multiple approaches. Dysbiosis could be alleviated by probiotics, and there are a number of studies that show positive data, but there's not really agreement on which antibiotic would be, which probiotic would be the most appropriate. Antibiotics, especially non-absorbable antibiotic rifaximin, which has been used a lot in patients with cirrhosis and has a good safety profile, could be used to reduce leaky gut and target specific bacteria in the gut. Prokinetics, as well as agents improving the gut barrier, such as FXR agonists, could also improve this. TLR antagonists could block TLR-induced signals that promote tumors or tumor-promoting reactions such as inflammation, fibrosis, proliferation, and anti-apoptosis. However, long-term use is problematic, especially because of systemic immunosuppression in these patients which are already immunosuppressed, and these drugs are currently not FDA approved. And in theory, it's also conceivable to directly target bacterial metabolism, so metabolites that promote cancer could be inhibited. How can this be realized in patients? One could consider primary as well as secondary HCC prevention. In my opinion, it would be important to identify high-risk patients. There are a number of ongoing clinical trials using some of these drugs that I mentioned before. However, rifaximin, norfloxacin, and FXR agonists, although they're given to patients with cirrhosis, the trials do not list HCC as either primary or secondary outcome, and there's only one study using probiotics. So overall, this is something that hasn't been realized in patients yet. Finally, I want to talk a couple of minutes about the microbiome in cancer treatment. There's very exciting and accumulating data that the microbiome promotes anti-tumor or regulates anti-tumor therapy in mice. So studies showing effects on chemotherapy and immunotherapy. Basically, what's been shown that antibiotics treated and germ-free mice have reduced effects of chemotherapy and immunotherapy. And soon after these mouse studies, there have been also studies in patients, and this is a rapidly expanding area, just showing these 3 landmark papers that were co-published in Science, all showing a role of the gut microbiome in immunotherapy in different tumors. And just one example for one study here showing that patients taking antibiotics have worse prognosis, patients with non-small cell lung cancer, as well as patients with immune responses against specific commensals also have these responses affect survival. So overall, there's strong data in mice and patients that the bacterial microbiota controls the tone of the immune system and increases anti-tumor responses. Finally, I also want to point out a new area of development, which is engineered bacteria as a therapy for cancer. This is a study here from Science Translational Medicine showing that bacteria can be used as a vehicle for therapy because they are strongly enriched in tumors versus other tissue. The liver here, not showing bacteria where these fluorescent bacteria are strongly seen within the tumors. And a second study showing this, also published in Nature Medicine, where they used bacteria that expressed a CD47 nanobody. All the bacteria, again, were strongly enriched in tumors versus liver or spleen. And the bacteria that expressed the nanobody against CD47 achieved a strongly increased survival in mice bearing tumors. So I want to summarize and answer some key questions on this topic. Does the microbiome have a role in hepatocarcinogenesis? So there's strong data in mice with multiple studies. In patients, the changes in HCC versus cirrhosis seem moderate, whereas there are strong changes in cirrhosis versus normal. And again, cirrhosis is a precancerous condition that can drive cancer development in the liver. A clear functional link in human HCC is still missing, and the role of the intertumoral microbiome as well as the microbiome need further investigation. Are there specific bacteria that promote HCC? There are no clear candidates as opposed to CRC, colorectal cancer. It seems rather that dysbiosis and the leaky gut drive this development. Is the microbiome a target for HCC prevention? Studies in mice suggest this, but this needs to be investigated in clinical trials. And finally, is the microbiome a target for HCC treatment? There are promising approaches combining the gut microbiome modulation, such as FMT, together with checkpoint inhibitors. This is mostly in non-HCC studies. In HCC, we first need to investigate whether microbial profiles correlate with the success of immunotherapy. And finally, engineered bacteria that grow specifically in tumors provide a new exciting opportunity for therapy, but this has only been done in mice so far. I would like to thank you for your attention.
Thank you, Robert, for an outstanding presentation. As always, we'll wait for a few minutes to take all the questions in the discussion. And I have the distinct pleasure now to invite Professor Suk from Korea, from the Halim University College of Medicine. He is a professor in internal medicine, also of the head of Microbiome Center and Institute for Liver and Digestive Diseases. And I had the pleasure of working with him for a short time. So he will be presenting a case, Dr. Suk. I hope it is not very difficult for us. All right, please go ahead.
Thank you for your introduction. I'm Kitaesok from Korea. In this presentation, I will summarize the previous presentations. I will present by disorder: alcoholic liver disease, non-alcoholic fatty liver disease, hepatocarcinogenesis, and targeting the gut microbiota. Liver disease is a clinical spectrum of disease that includes fatty liver, hepatitis, liver cirrhosis, and hepatocellular carcinoma. Most common etiologies are virus, alcohol, fatty change, and gut microbiota. In case of hepatitis, 7% of patients shows the mortality rate during 15-year follow-up. Regarding liver cirrhosis, 5-year survival rates are very low. Human gut microbiota is a complex ecosystem, consists of more than 2,000 species and more than 1 kilogram. Various factors such as lifestyle, genotype, physiology, and host immune system affect to microbial variation. Alcohol abuse causes intestinal dysbiosis and bacterial overgrowth. Certain gut bacteria can metabolize tryptophan into indole compounds that activate the aryl hydrocarbon receptor. Activated aryl hydrocarbon receptor induces the expression of interleukin-22 in lamina propria immune cells, which stimulates mucosal defense via the production of antimicrobial peptides, such as leg-3 beta and leg-3 gamma. Intestinal deficiency of leg-3 beta and leg-3 gamma increases bacterial translocation through the portal vein. In the liver, viable bacterial and microbial products induces hepatic inflammation, hepatocyte death, and fibrotic responses. In the non-alcoholic fatty liver disease, gut-derived pumps goes to the liver through the portal vein and recognized by toll-like receptor. The activation of toll-like receptor results in steatosis, cell death, inflammation, and fibrosis. In non-alcoholic fatty liver disease, insulin resistance, macrophage recruitment, and adipocyte dysfunction can be modulated by the intestinal-derived bile acid, LPS, and glucagon-like peptide 1. Dysbiosis and a leaky gut result in increased hepatic exposure to bacterial metabolites and MAMs. Bacterial metabolites such as bile acid ethyl, SEC, and CXCL16 dependent on NKT cell recruitment as well as hepatic stellate cell SASP. The contribution of bacterial metabolites TMA, TMAO, and ethanol to hepatocarcinogenesis is not yet understood. Bacterial MAMs such as toll-like receptor 4 against LPS and toll-like receptor 2 against LTA act on hepatic stellate cell to promote hepatic stellate cell SASP and the secretion of hepatomitogen epiregulin. MAMs may act on the macrophage to trigger tumor-promoting inflammation as well as hepatic stellate cell activation and fibrosis, which may promote hepatic stellate cell development. We cannot change our genome, however, we can change our microbiome. We can use FMT, probiotics, prebiotics, symbiotics, and engineered bacteria for the treatment of liver disease. We can use phage therapy, antibiotics therapy, and antifungal agent for the modulation of microbiome. FXR agonist and FGF19 analog can be used for the treatment of liver disease. Regarding FMT, we did FMT for the decolonization of carbapenemase, producing carbapenem resistant to Enterobacteria. We did a colonoscope and we used the Progen stool. In the result, the colonization rate within 1 month is roughly 45%. The colonization rate within the 3 months, roughly 75%. And the 6 months decolonization rate is 100%. Conclusion: in the liver disease, gut microbiome played a central role in the pathogenesis. Modulation of microbiome might be effective in the treatment of liver disease by regulating gut-liver axis. Thank you.
Thank you, Dr. This is enough. There's a lot of presentations that were done today. Very nice presentations, and thank you for this. And now it's time for discussion. Dr. Schnabel, we welcome you despite this very early hour in California. It's 5:40 a.m. there. So Robert Schwabe and I are still waking up. It's still 8:41 our time. But thank you everyone for this wonderful discussions. And please, to the audience, put your questions in the chat. Bernd, there were some questions for you in your response to your excellent talk. Dr. Varma said, in your opinion, what is the best strategy for improving gut wall integrity in patients with alcohol-associated hepatitis?
Thank you so much. I think the jury is out, and you know, there's certainly in patients very good approaches with, you know, you can use non-targeted approaches like with probiotics. I think the overall response from probiotics are very good. The only problem with the clinical trials is that none of the clinical trials has been reproduced. So I think this is something we need to pay attention to, you know, to clearly reproduce the same clinical trial with the same probiotics, with the same regimen, with the same length of time, length of treatment, to see if all of these results are reproducible. So probiotics are probably one of the you know, treatment options that could be used in the future to treat patients with alcohol-associated liver disease. Now, the more newer advanced techniques, like really where you have this precision medicine approaches, they are really in the preclinical stages at this point. We can't really make any judgments how this will affect our patients eventually.
Thanks for that answer. Anyone from the panelists want to comment as well? Dr. Sareen, Dr. Shastri, Dr. Yang.
Yeah, may I ask for
your,
yes. Probiotics. So what type of probiotics are we talking about, and what would be the dosage? Does it matters? And what do you think? Because we have a lot of probiotics on over the counter, and people talk about this, and just don't really understand whether there is any suggestions on what to be used and how much.
I don't know about Bernd's experience, but at least we do not use probiotics for treatment. At least we do it as part of research trials, etc., in patients at this stage, because as you currently pointed out, unless it's a pharmaceutical grade, we don't know what's actually in them. And there are certain analyses that have been done, at least in patients with pancreatitis, that probiotics in critically ill patients could actually promote higher death, because the critical illness, nothing to do with the underlying liver disease itself. But therefore, I think, especially with alcohol-related hepatitis, where, you know, antibiotics, etc., and a whole lot of other things, you really want to be very careful what you're putting into that patient. We don't use it short of a research study, but this is important to know that those are the few methods in which you can positively, potentially modulate the gut microbiome.
Charles, I completely agree. I mean, I might have phrased this in a way that this is clinical practice. The question I answered was, how can we stabilize the gut microbiota, and how can we stabilize the gut barrier? So in this case, one of the options is probiotics. I completely agree with Charles that we are far away from using this routinely in clinical practice. And so more clinical trials, as I said, you know, are needed to confirm the beneficial overall trial, the real good beneficial clinical outcome in these clinical trials. We will hear a very nice presentation at AASLD about this in alcohol-associated hepatitis.
Yes, because the problem is there is a lot of commercially available over-the-counters, and there is a lot of different brands and claiming. And I think that once the terms probiotics comes out from very good scientists like you, then the public might be misled, because there is no actually a brand for the probiotics. And I think sometimes as a responsible professional body, so we might need to clarify these issues, because I always encounter patients coming with bottles of probiotics from US, Canada, whatever, China. And this is sometimes very difficult, especially then they link the two things together, and we are far from that.
Dr. Lau, this is an amazing question that you actually raised, because there is clinical practice is very, very different now. And especially when you talk about traditional medicines in India as well as China and other places, they have such a huge impact on liver health, or at least perceived benefit versus not lack of benefit. So I think our message is consistent that, right now, more trials are needed and continue current management for at least alcohol-associated hepatitis. There are Dr. Sareen, Dr. Shastri, any of our Korean colleagues, any comments on this important
on
point?
this? Thank you. I would like to say that for alcoholic hepatitis, we finished one trial, what Dr. Shastri showed, where steroid eligible versus FMT was tried, and we had reasonably good response. In fact, a shade better than what we had with steroids. And the way it was that every patient, the donor was screened very carefully for enteropathogenic and all those, the complete protocol, and was on a standard diet. And the patients were followed up carefully. But I agree with you, it is still part of a clinical trial, and a senior resident has to be on watch all the time. You can't give it to everyone. Therefore, I would say there is a promise. It can be tried, and we use the nasoduodenal route rather than the other route. So in nasoduodenal, we feel that a small amount can regurgitate back into the stomach. So we have to be extra careful in that. But with that, this is a protocol that is being done at our place regularly. We would have done 200 plus patients, but a lot more needs to be learned. As far as the integrity of the bowel is concerned, so all the mate cells related studies, including zona occludens and others, were done. I don't remember them well, but it did change the mate cells. So what we used to do was to take a biopsy if the patient did not have coagulopathy while the jejunal, nasojejunal tube was placed, and we were able to see if there was a change in the circulating and other mate cell markers. But the field is in infancy, and we have to be doubly cautious. Thank you.
Another nice question by Dr. Pratibha Kale. Candida species are also commensal microbiota. Is there any other species than albicans associated with alcohol-related liver disease? Question is for Bernd, and after Bernd, everyone can answer.
We have seen, and we have published this, there are other species, and we are currently working on them. So the jury is out. It's probably, Candida is, from a proportional perspective, but also from an absolute abundance perspective, probably the most dominant microbial or the fungal species that takes over in patients with alcohol-associated liver disease. There are less abundant fungi that are also increasing. And like I said, we have very early data, so I can't, preclinical data, so I can't really say if it's associated with worse outcome in preclinical models. But yes, there are potentially other fungi that promote alcohol-associated liver disease. And then you also have the questions, and that's certainly something we have to also consider. Are there fungi, are there beneficial fungi, right, that are decreased during alcohol-associated liver disease? Not only alcohol-associated liver disease, but for example, NAFLD and NASH that are decreased during chronic liver disease, and this decrease of this beneficial fungi might also contribute to disease. So that's also something, that should be looked at. Thank you.
Yeah, and in patients with cirrhosis, again, we find an expansion of Candida in patients, and with and without antibiotics. But with antibiotics, the fungal diversity collapses and the Candida takes over. So I think it's a resident, it like any bug, it can be commensal under the right circumstances, but when it takes over, it potentially could cause an issue. And especially since we realize that of the fungal infections, the #1 cause was Candida also in patients with cirrhosis. Of course, it was not the same people that we took the ITS sequencing and did the infections off. When that quote unquote overgrowth or high abundance translates into an actual infection, like the bacteria, is a balance between local immunity, systemic immunity, and the virulence of the microbiota itself. So yeah, clearly, any other comments, Dr. Sareem, Robert, Shawley, Dr. Joe?
Not really, no.
Hello, I want to just make the one comment that on the previous question about the gut wall integrity in alcoholic AH patients. Dr. Lau mentions the number of the probiotics. We cannot really, many patients cannot really determine that. And I think the most probiotics is currently available is mostly, we call the grass in Korea, and the lactose or bifidose. But the gut microbiome has a huge, very different diversity species. So our lab published last year or 2 years ago about one of the gut microbiota called Rosebaria. The flagellin of the Rosebarias increased the gut tightness, increasing zone 1, particularly the proteins, and through the TRF5, the receptors. I'd like to let you know that we are currently doing some clinical trial in the 3 hospitals in Korea. So it's, I think, very early stage right now. So I think as the science advances, maybe a number of different options, or either the gut microbes or the particular cellular component or the metabolite may be used as a treatment to increase the gut tightness.
Thank you very much.
Can I ask the moderator one question and then one to the panelists? I wanted to ask
moderator. Well, I thought I was clean free. My God. Okay.
And maybe everyone can guide. And the question is, you have in alcoholic hepatitis, you give steroids and you do Lille score at day 7 because you think the response to steroids and change in the inflammatory markers would come in 7 days' time. However, when you give newer therapies like anakinra or maybe FMT is given at least by us for 7 days. So a Lille score at 7 days is unlikely to change. And we know that the engraftment or establishment of the flora takes at least whatever little studies we have takes 3 to 4 weeks time before they struggle to settle down and make some impact. So I have 2 questions to maybe Jess and Bernd and others. First, in a patient who has alcoholic hepatitis, how much time do you think it takes for the bacteria to get engrafted or established? And 2, should we not change from Lille to another scoring system where we assess maybe at 4 weeks or after that, or like delta MELD or a new score? Like, these are the 2 questions. Yes.
Okay, very important questions. And it tells you that, as things change, as life changes, as what you're introducing new therapies, we want to move with the time. And And LIL is actually more a score of futility rather than a score of prognosis itself. It's like stop the steroids if this doesn't happen. And it is interlinked with the steroid therapy. And if you are doing something that is in addition to steroid therapy, or in patients like you had published before who were not eligible for steroid therapy, then it really is a prognostic thing, but it's not an absolute thing. So depending on different studies, the engraftment rates are so different. And as you know, even in Clostridium difficile, Clostridioides difficile, where there is the most reports, you don't need to have full engraftment for the patient to actually feel better, get adequate outcomes. So as long as we stick to an outcome that everyone agrees, death, worsening of liver disease, etc., I think we'd be okay. When we talk about engraftment, even in FMT, as I said, in places where anybody's FMT will be better, like in C. difficile, all you need is a person who does not have FMT, doesn't have C. diff. If you put their stool, the patient will more than 95% get better, provided nothing else has happened. Even in those patients where there's a clear-cut improvement within days, the engraftment is either not there or it's not completely there. But the clear thing is what we want to talk about here is not to be too invested in the engraftment part leading to this. We want to go and make sure that we are doing our best. Like your protocol for 7 days, these are very sick patients. You want to give them the best and give them not one enema and then forget about it like we had done in an outpatients. Outpatients are a completely different ballgame. This person, you need to hit them and hit them hard if things need to be changed. And I agree with you that if your treatment lasts for 7 days, you cannot use a clinical scoring system that reads out in 7 days. That was actually related to steroid therapy, which has nothing to do with what you're doing. So it's not like that system is wrong. It's just that it may not be applicable to the greatest extent here. And you may want to also focus on things such as bile acid change profile, because we have found that, and the situation in alcohol hepatitis is very different, and Bernd, please comment. But the people who have restoration of their secondary bile acid producing capability in cirrhosis are the ones in whom the FMT sticks and it works. And in ones who don't, they don't. We had published this in JCI Insight with the oral capsules. But again, this is a very fraught topic and very important topic because these patients are so sick.
Any comments, Bernd, on this?
No, I agree with Charles. I think we are a little bit far away, to really know what is, and this was Charles mentioned, what is the real best readout for eventually who is getting better? Why is that patient getting better? So in other words, if we go towards this, what we always say, or this precision medicine approach, right? So looking at metabolites, looking at other factors that improve in the patient, that improve with the engraftment, and then link this to the FMT and to the patient. For example, short chain fatty acid. For example, like Dr. Bajaj mentioned, bile acids. Would this be the better follow-up markers, biomarkers for getting, or potentially even, markers for improvement? So causing causality in terms of improving liver disease. Would these be the better markers than only looking at like engraftment? So I think, the more we learn in the future, I think the more precise we can get. And then we can use away from FMTs, this is future, obviously, more to, for example, bioengineered bacteria, synthetic bacteria, like postbiotics, like metabolites that we know might be sufficient to do this, if it's feasible, or maybe a combination out of many of these factors. Thank you.
Okay, thanks. I have a brief question to Dr. Koh, and maybe after that, Robert can address that. Do you think and it may look stupid as a question, do you think the change in bacterial flora in NAFLD is a result of hepatic disorder, or is the cause of hepatic disorder. And how do you address this question? Every day people keep asking the cause and effect, and the same will be true, maybe Robert can address, is the development of carcinogenesis in the liver. Is it bidirectional or unidirectional?
Yes, I think that's the very important and questions, and also I think that every time we do the some of the patients, the data, is this the correlation or causalities, and or using the different types of models, including cell or germ-free or conventional mice, we're trying to demonstrate the causality of the particular microbes. I think dysbiosis is very the vague terms when as a microbiologist see dysbiosis more like in terms of ecologies. But I believe among these dysbiosis, I believe that particularly the strains of the microorganisms or the molecules of microorganism or metabolism of microorganism is the responsible for these some of the pathogenesis. So I believe we trying to identify that and then demonstrate in the causality using my animal models and so on. So from my talk, I adapted these models. We trying to identify the number of different markers, and actually I show one of the isolate, but we screen over the 100 the microbes trying to identify the particular strains of microbes to demonstrate the causality of the elevation of the NAFLD models in mice model in this case. And so I think we need to look at very closely that, but I believe the many studies the report the causalities of the particular strains or molecular mechanisms. So that's what we are looking for.
Yeah, so if I can quickly comment from my perspective, yes, it's a two-way bidirectional relationship, and obviously in HCC there seems to be a component driving this. The intratumoral microbiome that I have briefly mentioned is something that we really don't understand very well, especially in HCC. There need to be more studies. But overall, I think the biggest gap that in the area of HCC is really the understanding of therapeutic responses, and I think melanoma is really driving this. I mean, like with the immunotherapy, the HCC field is following. So we really have to understand much better what the role of the microbiota is in dictating therapeutic responses, and this may be a really important area that could be modified. As I mentioned already, also I think that the engineered bacteria as a therapeutic vehicle is something obviously that's very far away from the clinic, but the mouse studies are extremely exciting. So for the HCC field, which is different from cirrhosis, I think we need to shift our attention a little bit.
Yeah, if I can comment here, like in severe alcoholic hepatitis, like there must be some trigger from the gut. It is the gut which is causing the alcoholic hepatitis. A person who is been drinking for a long time suddenly develops a hepatitis or alcoholic hepatitis flare. So he's been drinking and he's been eating same forever. So what is the trigger for the onset of alcoholic hepatitis? Can it be gut microbiome or any particular change in the microbiome?
I think you have a point, Dr. Shastri, and the issue is wide open, but in the interest of time, we have two questions. One, I'll come back to you, and one to Bernd, Robert, maybe how do you use cytolysin and what dosage? One of the questions there is, how can we dose the cytolysin? Or anyone else in the panel?
I think this was in response. Yeah, Bernd, this was in response.
Sorry.
You had said the cytolysin thing. I think that the attendee was not clear as to what you meant by the cytolysin. That is, yeah, just
yeah. So cytolysin seems to be, first of all, a very, very good biomarker for disease outcome in patients with alcohol-associated hepatitis, so with alcoholic hepatitis. And this was based on a qPCR test from patients where we took the stool and essentially did the qPCR for the 2 subunits in the stool from these patients for the cytolysin. So we would propose that this is and probably very specific good biomarker for alcoholic hepatitis rather than any other disease, at least in our hands. And obviously this needs to be confirmed by other groups, by other studies who repeat this. So this is the first part, very good biomarker seems to be the case. And then for treatment, we are far away, but it certainly can be proposed to use a clinical trial targeting these bacteria that make the cytolysin with, for example, bacteriophages. I can also envision, other antibiotics that could eradicate these bacteria. The problem with Enterococcus faecalis is, and we know this from our whole genome sequencing, is that you have many, many, many antibiotic resistance genes in this box. So that might be a step which we have to overcome if you want to treat these patients with antibiotics. Thank you.
Yes, thank you. I think the time that we had is too short for such a big and hot topic.
Yes.
May I ask Jess to kindly give some more comments before I conclude? Jess, your wisdom and your wise comments.
Oh my God. Okay. Thank you so much to APASL, and thank you for this wonderful panel of speakers and my distinguished co-moderator and the president of APASL. This is the who's who of APASL is represented here, and who's who of world microbiota research in liver disease is represented here. I think there was an excellent talk, even though many talks that we concluded are not ready for prime time yet. It still gives us a lot of food for thought, not just to us, but to the more than 100 attendees that were here and were actively participating and listening. And I think this is something that APASL should think about much more carefully, and maybe do 2 hours next time so that we have a lot more interaction and a lot more junior investigators involved in presenting cases and already into the program. So it's a good problem to have. There is too much to be done and actually things to have, rather than sitting and no one interacting. Well, thank you so much for your time. I have to run to clinic. Okay. Take care.
Thank you, Jess. And I would like to thank our president, Professor Jinmo Yang, and the whole Korean group, and Dr. Suk, particularly, who is a pillar in Korea in the microbiome area, but we could not discuss more. And I do say that these are very young people, including Dr. Shastri, Dr. Suk, Dr. Ko, and of course you, Jess. Thank you all. And I would like to say that this is one of these sessions, and I hope the annual meeting, which is going to be held in Korea, would have a full day or longer time for this. Koreans have extremely high standards of microbial research, so we will have more of them. But the APASL, as one of the representatives from APASL, I would like to thank you. Thank our other panelists. Are there any other comments from our panelists? Sorry, I didn't ask Professor Yokosuka, Professor Kanda. Sorry, any other comments before I conclude, Dr. Kanda? Dr. Yokosuka?
Yeah, I enjoyed this, meeting very much. And I think we have to think about a risk of using the phages or such sort of things. But anyway, I enjoyed this meeting. Thank you very much.
Dr. Kanda?
I also enjoyed this meeting. Thank you very much. everyone.
George?
It's wonderful meeting, especially now we are going to immunotherapy for hepatocellular carcinoma. There is a lot to understand. And
Yes.
congratulations to the work of the Koreans and all the speakers and moderators.
And Professor Lim?
Thank you very much, Professor Sarin, and all the speakers and the chairpersons for making this webinar to be wonderful. Thank you very much again.
You and Professor Jinmo Yang are the pillars for this webinars, and we thank you and your team. And with that, I would like to say profuse thanks to all our speakers, especially for getting up in the early morning and being with us. It's a huge effort, and APASL and all of us deeply appreciate. And we hope to be together in near future. Thank you all. Bye-bye.
Thank you, Professor
Thank you
Yang.
very much.
Thank you very much.
Bye.
Bye. Thank you.